Hello everyone, welcome to the second Women in Bioinformatics Data Science Latin American Conference. My name is Cynthia Castillo-Silva and I'm here to talk to you about the spatial variability of SARS-CoV-2 Spike protein and its effect on COVID-19 disease outbreaks. In late December 2019, a different strain of coronavirus which caused severe acute respiratory syndrome emerged in Wuhan, China. COVID-19 disease caused by SARS-CoV-2 quickly was spread to other countries, and the World Health Organization declared the COVID-19 outbreak as a global pandemic on March 11. Transmission occurs via inhalation of respiratory droplets exhaled from an infected person. SARS-CoV-2 infects type 2 pneumocytes localized in the pulmonary alveoli. As the virion binds with the ACE2 protein, another membrane protein cleaves the spike proteins at multiple sites. These mechanisms allow entry of the virion, releasing its RNA strand. However, the mutation rate for RNA viruses is drastically high and provides a competitive advantage for viral adaptation. Clade systems allow us to group these mutations by shared characteristics. In the screen, we can see the most known clade system for SARS-CoV-2 taken from GSAID's database, where we can see distributions per region and per clade for all the samples. In this work, we will not use the clade system. We will instead perform a random forest algorithm to classify all mutations into characteristic groups. The data we used was from the NCBI. More than 39,000 complete spike sequences were retrieved from its database. It is important to note that the distribution of the samples was not equal. Almost 90% of the data was taken from the US and Australia. Once we get all the samples, we proceed to align these samples. After this, a PAM2 matrix was used to define a metric to score amino acid mutations in these proteins in order to determine their evolutionary divergence in phylogenetic trees. Then, we divided our samples per region to perform a topological data analysis to find out how different from the regions were from each other. From the heat map, we showed that detected cross-correlations between pairs of datasets. Particularly, we can see 4 important clusters in here. In this part over here, this one, this other cluster, and this other one, in these zones. Also, the TDA is a robust method for n-dimensional analysis. Machine learning analysis is better at discriminating variables and finding pattern characteristic mutation clusters of the protein sequences were classified using a random forest algorithm and diffusion maps. This allowed us to identify the most important mutation patterns related to outbreak rates in different geographic regions, as we can see in the figures on the screen. It is important to keep track of the mutations that occur in the RBD domain of the S protein because of natural adaptation with the host. We found 5 hypervariable motifs here in these parts. More amino acid variations occur in regions of high content of NAG, as seen in the picture of the left in color cyan. Topological data analysis allowed us to identify close relationships between mutation patterns, suggesting roots of the spread of the pandemic. The random forest algorithm was applied to detect significant missense mutations, some of them already associated with increased infectivity and virulence. And finally, we identified the regions of high variability along the spike protein sequence, particularly localized in the NTD region that has high contents of NAG. This is relevant to assume that the NTD region is involved in evolutionary adaptation. Studying the variability of the spike protein will require significant efforts to develop wider spectrum anti-SARS-CoV-2 strategies. It is necessary to keep ongoing mutational sequences and monitoring studies, and to avoid aiming efforts to the high variability regions to contain the COVID-19 disease. I'd like to acknowledge Scientific and Technological Research Support Program, PICIT, of the UNL for supporting this work. And finally, I'm honored to introduce you to our research team. For further information regarding this work, please contact Dr. Francisco Hernández to the email shown in the screen. Thank you.